Here is second half summary of FSc Biology Part 1, Chapter 2 Biological Molecules for ppsc lecture and subject specialist test preparation. It is humble request to all of you, please watch the video online, don't download, it really hurts our effort and sign of discouragement. If you want to watch first half, press I button and go into description for the link. Now we will read proteins up to end. Proteins. Proteins form 50% of dry weight of cells. It means that if you remove all water from cell, the remaining dry weight of cell contain 50% proteins. They are structure builders in organism, means that all the structure in organism are built or made up of proteins. All enzymes are proteins. Hemoglobin not only transport oxygen but also lipids and metal ions. Antibodies are proteins which defend against pathogens. Proteins are polymers of amino acids. It means that the building block of amino acids are protein from few to 3000 AA in single proteins, which may be present in a chain, in a row. About 170 types amino acid have been discovered so far, and among these 170, 25 amino acid type are constituent of protein, that is, they are present in one or either way they are present in protein, while most are formed from 20 types. Please remember that most of protein are formed from 20 types of amino acids. In a amino acid and carboxyl group are attached to alpha carbon. Please remember to which carbon these two groups are attached. Please, it is very important. Glycine amino acid is single and has H atom instead of R or any CH3 group. So, glycine amino acid has H atom on both sides. Alanine has CH3 group. Amino acids differ in R group only, or you can also call it as side chains. Peptide bond is formed by removal of OH group from carboxyl and hydrogen atom from amino. Please remember, it is very important MCQ. Glycyl alanine is a dipeptide, means it has two amino acids. Properties of proteins are determined by number and sequence of amino acid in molecule. So, on which factor properties of proteins depend? Number and sequence of amino acids in that molecule. Primary structure. Sequence of amino acid in a protein comprises primary structure. That means that simple sequence of amino acid or linear structure is called primary structure. F.Sc discovered sequence of amino acid in protein first time. Please remember his name. Insulin has 51 amino acids in two chain as 21 and 30 amino acids individually held through disulfide bridge. Disulfide bridge, it is very important word to remember. Insulin in two chains are held through disulfide bridge or disulfide bond. Hemoglobin has 4 chains, 2 alpha and 2 beta. Each alpha has 141 amino acid and beta has 146 amino acids. Human body has 10,000 protein. In sickle cell disease, 1 amino acid in beta chain out of 575 is replaced. Glutamic acid is replaced by valine. So, if 1 out of 575 is replaced, the whole shape of the cell is changed. Secondary structure. Most common secondary structure is alpha helix. In spiral formation, it may be in a spiral form or in a form of spring. Each turn of helix has 3.6 amino acids. Very important fact, please open your eyes as well as ear. Hydrogen kept the structure together. Which structure? Secondary structure. Beta plated sheets are formed by folding back of polypeptide. Now, tertiary structure. Bending and folding of polypeptide upon itself form globular shape, which is tertiary. Globular means round or spherical type. Tertiary structure is maintained by three types of bonds, that are ionic, hydrogen and disulfide. Disulfide bond is between two adjacent sulfur atoms. Please remember it. Stable tertiary structure have hydrophobic amino acid inside while hydrophilic on the surface. Please remember this fact is very important in ppsc exams. Quaternary structure. Tertiary chains aggregate and held together by hydrophobic interaction, hydrogen and ionic bonds to form quaternary structure. These three forces are very important to hold tertiary chains to form quaternary structure. Which three one? Hydrophobic interaction, hydrogen ionic bond. Not all protein exhibit quaternary structures. Hemoglobin exhibit quaternary structure. Now, most important of the protein, fibrous versus globular protein. Polypeptide in the form of fibrils. Secondary structure is most important. They are insoluble in water. They are non-crystalline and elastic. They play structural roles. For example, fibrous proteins are silk, fiber, myosin, fibrin, keratin, etc. These were fibrous protein because they are in the form of fibers. Now globular proteins. Polypeptide is in the form of spherical or ellipsoidal information. Tertiary structure is important. They are soluble in water. They can be crystallized. They play functional roles. For example, enzymes, antibodies, hormone and hemoglobins. These are very important. Examples of fibrous and globular protein are very important according to MCQs in ppsc exams. Now, here is an important information. Keratin is present in nails, fibrin in blood clotting and myosin is present in muscle cells. The next is nucleic acid. These were first isolated in 1869 by F.Sc from nuclei or nucleus of pus cells. Please remember, nucleus of pus cells in 1869 by F.M. They were named nucleic because of discovery in nuclei having nucleus and acidic because of acidic nature. So they are two termed which are nucleic acid having nucleus and acidic nature. DNA. Deoxyribonucleic acid is present in nucleus, mitochondria and chloroplast in cell. RNA. Ribonucleic acid is present in nucleus, ribosomes, cytosol and other parts. These are very important MCQs. Nucleic acids are polymer of nucleotide units, means chains of nucleotide unit form nucleic acid. Each nucleotide is made from 3 subunit pentose sugar, nitrogenous base and phosphoric acid. Sugar in DNA is deoxyribose, while in RNA it is ribose. Nitrogenous bases with single rings are pyrimidines, while double ring purine. Pyrimidine are cytosine, thymine and uracil, while purine are adenine and guanine. Nitrogenous bases attach with first carbon of the sugar and phosphoric acid with fifth. Base and sugar together form nucleoside. Nucleoside and phosphoric acid form nucleotide. In deoxyribose sugar, one oxygen is removed from OH group at second carbon. Adenosine triphosphate is a nucleotide used as energy currency by cell. Adenosine monophosphate AMP has no high energy bond and adenosine diphosphate has one high energy bond ADP. Adenosine triphosphate ATP has two energy bonds between phosphates. DNA. DNA is basis and control the properties and potential activity of cells. There are 4 types of nucleotides which form DNA: DAMP, D-adenosine monophosphate, D-GMP, D-guanosine monophosphate, D-CMP, D-cytidine monophosphate, D-TMP, D-thymidine monophosphate. Nucleotides linked through phosphodiester linkage. Very important to note that which linkage upon which nucleotide link to form DNA. NAD is a coenzyme nucleotide adenine diphosphate, which is a dinucleotide. All nucleotide may have mono, di and triphosphate as MP, DP and TP, monophosphate, diphosphate, triphosphate. Like for example, AMP, adenosine monophosphate, ADP, adenosine diphosphate, ATP, adenosine triphosphate, and then GMP, guanosine monophosphate, GDP and GTP respectively. Now DNA. Erwin Shergraff in 1951 provided ratios of different bases. Adenine equal to thymine while guanine equals cytosine. Sources of DNA, adenine, guanine, thymine ratios are described in this table. Man 30.9, guanine 19.9, thymine 29.4, cytosine 19.8, in sheep 29.3, adenine, guanine 21.4, thymine 28.3, cytosine 21.0, weight 27.3, 22.7, 22.1, 22.8 in yeast, adenine 31.3, guanine 18.7, thymine 32.9, and cytosine 17.1. Wilkins and Franklin determined structure of DNA using X-ray diffraction method. Watson and Crick build model of DNA. Coiling of DNA is anti-parallel. These are very important facts to remember, that who build model of DNA, Watson and Crick, and the coiling of DNA is parallel or anti-parallel. 2 hydrogen bonds between adenine and thymine, while 3 hydrogen bonds between guanine and cytosine. There are 10 BP in each turn and 34 angstrom or 3.4 nanometer in length. Now the type of cell, amount of DNA, nuclei or in picogram in chicken and crab has been shown in the table. Red blood cell, liver cells, kidney cells and sperm cells, and their amount is very important. In E. coli, 5 million bases are arranged in linear order in DNA, which code for 4 to 88 proteins. First completely sequenced genome was of Haemophilus influenza and was published in July 28, 1995. It is very important MCQs and have already been asked in paper that which was the first genome, and that was Haemophilus influenza. RNA. RNA molecule is single-stranded and can fold on itself to give double helical feature. It's give double helical ring structure. RNA has uracil instead of thymine and is formed by transcription of DNA. mRNA, yani messenger RNA, carries genetic information from DNA to ribosomes to synthesize protein. For the synthesis of 1000 amino acid proteins, messenger RNA should have 3000 nucleotides. mRNA is about 3 to 4% of the total RNA in cell. Transfer RNA or tRNA is about 10 to 20% of cellular RNA and is small, about 75 to 90 nucleotide. It transfers amino acid to the site. There are 20 types of transfer RNA for each type of amino acids. Ribosomal RNA or RNA is major RNA, about 80% of the total RNA, and is part of ribosome. On the surface of ribosome, it interacts with transfer RNA to translate information. Now, conjugated molecules. Formed from different type of molecule like glycoprotein and glycolipids, most cellular secretions are glycoproteins in nature. Both glycoprotein and glycolipids are component of plasma membrane. Lipoprotein forms structural framework of membranes. Nucleic acid attract basic protein to form nucleoprotein. Nucleohistone are present in chromosomes. Nucleoprotein play important role in regulation of gene expression. These all are very important facts about conjugated molecules and have already been asked in ppsc exams. The upcoming video is of chapter 3, enzymes. So please connect it with us, subscribe, comment and share. Also press the like button. Thank you for watching and learning. Stay blessed. Allah Hafiz.